Hello, everybody, and on behalf of the workshop organizers, I welcome to this workshop. I'm very glad to see all of you here. I've seen all the faces, which made me very delighted, but also new faces. I'm looking forward to meeting. And basically, what is going to happen? This is the workshop, as you know, of the year, the emerging researchers in artificial life. That is the student chapter of the ISA, the International Society for Artificial Life. That is the organization behind the ALife conference, as Abe will detail later on. And as we can see from the schedule, what is going to happen now is that after this brief introduction, Abe will give us an introduction to ERA, its board, as well as a call for the elections for the upcoming year. Then we will move on to announce the 2022 ISAL Best Student Publication Award that this year went to an outstanding paper on how a phenotypic plasticity can stabilize evolution in dynamic landscapes, and here an invited talk from one of the two authors. After that, we will move on to the main track, we can say, of this workshop, which focusing on lightning talks, where some of you emerging researchers will showcase the research in brief lightning talks to have the spotlight upon themselves. Having said so, I leave the floor to my colleague Abe that will move on with the introduction to ERA. And we cannot hear you.
Oh, Abe, yeah, I can't hear you.
Let's wait for Abe to be back. So it looks like we're having audio problems in one of the rooms.
Am I audible now?
Yeah.
Oh, fantastic. All right. So as I was attempting to say earlier, I'm really glad to be here. ERA is the Emerging Researchers in Artificial Life, the official student and early career researcher group of ISAL. Our mission statement states that our purpose is to provide opportunities for students and postdocs, as well as other early researchers interested in artificial life to interact with each other, to develop and to contribute to the broader ALife community. Richard, if you wouldn't mind turning the slide. Thank you. So some of the things that we do, we maintain a welcoming network of early career researchers interested in ALife that's centered around our Slack, which I can send an invite link to in the Zoom chat in just a moment. We hold events like this one where people can interact and learn more about all the different sorts of ALife research, ALife interests that we're pursuing. We provide advice, informal mentorship for people interested in learning more about the ALife community. And we represent the interests of early career researchers on the ISAL board via our board representative, Richard. So the ERA board includes a number of other positions in addition to our ISAL board rep, we've got the general chair, the vice chair, the ISAL board representative, communications chair, equity chair, and conference chair. And I'm not going to get into all the details of what each of these positions does, but I'll quickly introduce each of the people who are here. So if you can move on, Richard. Thanks. So I'm the general chair for this year. I'm Leite.
Abe, we cannot hear you anymore.
Oh, I thought that you would be just very, very briefly introducing yourself and then so forth.
Oh, okay, I'm sorry.
Hey.
Yes, I'm Barbara, the vice chair.
Yeah, I'm Imi, the equity, the DEI chair.
I'm Federico, the conference chair.
And then we don't currently have a communications chair, but we are hoping to refill the position last year. The most recent communications chair was Austin Ferguson last year.
And then finally, it's me. I'm the ISL board representative. I'm Richard. Nice to meet you all.
So now you may be asking yourself, how can I join this august group of people? And well, can you? Yes, almost certainly. The only requirement is that you should be a member of ERA, and that's a very informal requirement. We would hope that you're in our Slack, that you have an interest in ALife, and that you haven't yet held a permanent position. There's a bunch of different things that if you're any of them, then you're certainly qualified, but you can also ask us if you're in any doubt. You can't, if you want to join our board, you should not currently hold another position on the ISAL board or be standing for the board election. So I want to, again, invite all of you to join, to participate as much as possible. And I want to quickly share some of the elections process and timeline with all of you. So the first stage is the nominations period, which can begin as early today as if you like, and will end in late September. Then we'll have a short voting period, and then finally the next terms will begin in mid-October. And to clarify the nominations process, in case any of you are interested, any ERA member may nominate another member for a position in the board by the timeline, and this, by the deadline, and this will be stated at later notice. And this nominations process will take place in the elections channel in the ERA Slack. Self-nominations are welcomed. All non-self nominations should be accepted by the nominee before they'll be officially recognized. Any additional member may second or third and so forth a nomination. This isn't strictly required, but it's a tradition and it helps show support for the nominees. The current ERA board will certify that any nominees are eligible for the role. So that's my spiel about the ERA board, and I'm happy to take any questions at this time. We're also,
Are you board members, are you all emerging researchers yourselves, or are you a little more advanced?
Yep, we're all early career.
Thank you.
I actually first joined the board as communications chair during my last year as an undergrad, so we're really a very, very open crew.
Wow.
And we're also really happy to take any other questions on the chat here or in our Slack later. All right.
So now we will move on to announce the best year publication for this year. And I leave the floor to my colleague Amy to announce the winners, as well as read the laudation from the judging committee.
Yeah, so thank you very much, Federico. So yeah, thank you. I'm glad to be announcing the winner of the 2022 ERA Student Paper Award. And before I do, I just want to say thank you to everybody who nominated the papers, and well done to everybody whose papers were nominated. We had an opportunity to review all of them, and they were all excellent. Just to briefly describe how the papers were assessed, the review panel chose to assess each paper on 5 different points on a 5-point scale. So scholarly rigor or scientific rigor, I can't remember the exact terminology, but it's effectively how well has the argument been made and presented? How well do the results and the discussion support the argument that's put forth? We weren't necessarily interested in what the hypothesis was or where it sort of stood in the grand scheme of things, but rather how well has the argument been made? Accessibility, how does it read? How is it presented? ALife is so diverse in our expertise and our backgrounds, and so how well does that paper, how good of a job does that paper do in being accessible to people of those different expertise and backgrounds? The student contribution, sort of self-explanatory, how much of a contribution do we perceive the student to have made to that paper? And that's usually derived through the author contribution statements in those papers. And then finally, the likely or the perceived impact both within ALife and outside of ALife. Like I mentioned before, ALife is so diverse in its backgrounds and expertise, and it's a wonderful playground to explore ideas that are also relevant to other fields as well. And so we assessed all these papers on their likely impacts, not just on our field and our community, but also those communities that might be adjacent to ours as well. So the good news is that all the papers scored very highly on these 5 points, and were a pleasure to read. But ultimately, there was one standout winner. Richard, sorry, yeah, and just to extract some of the comments from the nomination comments that we received. So we had comments like, this is an excellent research paper, leveraging the strengths of digital evolution to make important insights into the dynamics of life. A little more beyond the interesting results, the authors have done an excellent job of making their work as accessible and reproducible as possible. In particular, they have all of their code and data available and beautifully organized, ensuring that others will be able to build off this work easily. 3 out of the 4 authors are or were students within the last 3 years, and that this article represents a truly fantastic effort and result, regardless of the research stage, making it all the more impressive. And also, we believe that this nomination represents sound science with novel results that are of relevance to the artificial life community. And yeah, Richard, just to share some of the comments from the reviewers as well of the papers, Richard, thank you. So all the reviewers commended the scientific or scholarly rigor of this paper. The reviewers were all impressed by the accessibility, mentioning that the paper was coherent, easy to follow, that there were adequate descriptions of the experiments, the results, and the discussion that were all presented to a very high standard, making it accessible and easy to read and comprehend for people, for whom this particular area was not their expertise. And there was general agreement that this work has the potential to be impactful both within the ALife community, but they also saw it potentially inspiring applications and future theoretical work in other disciplines as well. So without further ado, I'd like to present the winner of our student paper, Richard. Alexander Laleni and Austin Ferguson, who I believe is going to be presented today on and the other authors as well, Grant and Ofria. The adaptive phenotypic plasticity stabilizes evolution in fluctuating environments. Now, and perhaps some of you had an opportunity to see Austin's talk this morning on this, and I'm quite excited to hear him elaborate on this a little bit. So the floor is yours, Austin. Well done. Well done.
Thank you. Y'all are too kind. Thanks again. This is great. You already mentioned this. I did give this talk, more or less the same talk, like an hour ago or an hour and a half ago. Apologies if you were in that session and then you have to come listen to me again. They're similar. This one has a little bit more detail. But yeah, thanks again. And also, I want to stress, so Alex would have been presenting this, but he caught COVID like last week. So I'm tipping in, and Alex always gives absolutely phenomenal presentations. So sorry you're stuck with me, but we'll try to make the best out of it. Can you all see my screen? Awesome. All right, so let's go ahead and dive in. As you mentioned, this was work done with Alex Lalagini, who was a PhD student at the time. This was actually the last chapter of his dissertation, so he's now Dr. Lalagini. And Kruma Grant was a postdoc at the University of Idaho at that time. So, you know, we did have some early career. Oh, and I'm a PhD student, I should have mentioned, at Michigan State University. So yeah, it was a pretty young effort. And then we also had Charles, who is Alex and I's PhD advisor. All right, I'm gonna start my timer real quick because I forgot to prep that beforehand. Give me just a split second to make sure I don't ramble too long. All right, so let's dive in. Our paper was titled Adaptive Phenotypic Plasticity Stabilizes Evolution in Fluctuating Environments. All right, so as mentioned, this was published last year in the Frontiers in Ecology and Evolution journal, specifically on this special issue on digital evolution, looking at how we can use digital evolution to grant insights to biologists. And I bring this up because there's actually another talk from the special issue at ALife this year, which I'm really excited to go to. It's on symbiosis, and I think it'll be really good. So strongly recommend y'all check it out too. But okay, to talk about this particular paper, first we need to talk about phenotypic plasticity. And I'm actually using this quote from West Eberhard here, or this definition. So they define phenotypic plasticity as the capacity for a single genotype to alter phenotypic expression in response to a change in its environment. And if we think back to the title, we're specifically talking about adaptive plasticity. And just as a really loose, easy definition, I'm going to say that adaptive phenotypic plasticity is just when the plastic response is beneficial to the organism, because it could be non-adaptive or maladaptive, but here we're solely focused on adaptive plasticity. All right, so all of us as authors were really interested in the evolution of phenotypic plasticity. And when you dive into that literature, there's a lot of work that's been done on the subject. So both in like an ALife and digital evolution, neuroevolution stance, but then also on the theoretical side and the biology side, there's just a ton of work on the evolution of phenotypic plasticity. It's a nice, rich literature. However, for this particular paper, we were interested in what comes next, right? So if you evolve phenotypic plasticity, how does that affect future evolutionary dynamics in the population? So there's a good bit of work done on this topic, but it's mostly theoretical or math models. It's really hard to do experimental evolution, especially in wet lab systems, because there's so much that you have to control for that really makes it intractable. And it's also hard to find natural systems that hit just the right requirements to allow you to do these tests, right? So we decided to do what we always do and use digital organisms. So in this case, we're talking about the AVITA digital evolution platform. And the only thing that I really need you to know is that organisms are self-replicating computer programs. We often visualize them like this, where they have a genome, which is just a bunch of these really, really basic computer instructions that don't do much on their own. But when you combine them in an actual genome, they can form some pretty complex behavior. And the other thing I guess I want you to realize is that, they take input and put out some output. So in our case, we give them some numbers, and if they output the right tasks, and in this case, the tasks aren't super important. They're, really basic bitwise logic tasks. But if they output them correctly, we reward those organisms. So they get an advantage, they get more CPU time compared to the other members of the population. So for this particular paper, what we did was we set up 6 different tasks. And to create this fluctuating environment, we split these tasks into 2 groups. So we have group 1, which is tasks 1 through 3, and then group 2, which is tasks 4 through 6. And then we decided to make 2 different environments using these groups. So environment A rewards tasks 1, 2, and 3, but it punishes tasks 4, 5, and 6. So if you do this task, we reduce your CPU time, and you're at a disadvantage, compared to the rest of the population. And then environment B is the same, only completely mirrored. So, we inverse the rewards and punishments, and now tasks 1 through 3 are punished and tasks 4 through 6 are rewarded. And then we take these 2 environments and we create this little world where it fluctuates perfectly between these 2. So there's a set amount of time that you're in environment A and then environment B for the same amount of time, so on and so forth through the entire experiment. So now that we have this world set up, it allows us to create our 3 experimental treatments. So we have a plastic treatment that's in this fluctuating environment that has one more key piece. All we need to do is allow the plastic organisms to sense what's being rewarded, or effectively what environment they're in. So if they can sense what's in the environment, then they can actually tailor their response, their phenotype, to that environment. We can compare this to a non-plastic treatment, which is in the same fluctuating environment, but we don't give them those sensors. So since they can't tell which environment they're in, they can't, tailor their phenotype. They can't express different phenotypes in different environments. And then lastly, we have just a third static baseline where they don't have sensors because the environment's always the same. It always rewards all 6 tasks. And then one last thing to mention is that we do precondition the populations. So for each experiment, we run 2 phases. And in the first one, we do evolution, just like we do for phase 2, for the same amount of time. But this preconditions the populations to whatever environment they're in. And this specifically matters for the plastic populations because we want to make sure when we're doing our analyses, we're testing actual plastic organisms, right? So we run phase 1, we find the most abundant genotype at the very end of phase 1, and then we plop it in as the ancestor for phase 2, and then run, another 200,000 updates or 30 to 40,000 generations. And it's in phase 2 that we do our experimental controls and actually conduct our analyses. So now we can actually get into our research questions, with the first one being, does adaptive phenotypic plasticity influence the speed of evolutionary change? So to get into this, we need a way to quantify evolutionary change. And to do that, we do a lot of phylogenetic and lineage analyses. So thankfully, Avita spits out at the end of the run a phylogeny that stretches from the ancestral organism all the way to all the extant organisms at the very end of that phase. So we can trace back from any organism and see all of the parents until we get to the ancestor, which is really nice for some analyses. And then for others, we want to look at a particular lineage. And we decide which lineage to look at by looking at the extant population here on the right. And we count how many organisms have each genotype. So then we decide, okay, we're going to consider the most abundant genotype or the dominant genotype. So in this case, it's this 25, this green color here, and we can extract its particular lineage. And thus we end up with this nice sequence of genotypes from the ancestor all the way up until this most abundant genotype. All right, so now that we have our lineages that we can look at and our phylogenetic trees, we can start looking at the actual data. So all of my plots here are going to be the same format. So on the x-axis, you have the static treatment, and then the non-plastic treatment and the plastic treatment. And then on this particular plot, the y-axis is our mutation count. So we're looking at how many mutations accumulate along the dominant lineage for each replicate. So as we look at the data, each dot here represents one replicate. So we see there is a significant difference between them, but static and plastic treatments have pretty similar mutation accumulation, especially when we consider how much greater the mutation accumulation is in this non-plastic lineage here in the middle in the light blue. So, thinking about this, it makes sense because while the static and plastic populations don't have to continuously change their genotype because the environment doesn't change in the static condition, and in the plastic condition, you're plastic, you can handle the different changes without changing your genotype. Whereas the non-plastic populations, every time the environment changes, you have to rely on mutations to update your phenotype to match the current environment. So this checks out. And then we can also look at other metrics, right? So here's phenotypic volatility, which is if we look along the dominant lineage, how many times when we go from parent to offspring does the phenotype change? And we do take into consideration like your phenotype's always changing in the plastic one. So we're looking at across both environments, when you concatenate those results together, when does your phenotype change? And here we see similar results. So we see really low phenotypic volatility. Again, this is a log scale. It's really low phenotypic volatility on both the static and plastic treatments, and considerably higher volatility on the non-plastic treatment. So the phenotype's changing rapidly in the non-plastic treatment compared to the static and plastic. And then, like I mentioned, we can also look at the whole phylogenetic tree instead of just the one lineage. And here we're looking at coalescence events. And by coalescent event, I just mean if we look at any point in the run, Avita can look at all the extant organisms and just say, hey, where is their most recent common ancestor as we trace back the phylogeny? And every time that that most recent common ancestor change, we log that as a coalescent event, which is really a selective sweep, if you're familiar with the term, because now all the organisms in the extant population are descended from that new most recent common ancestor, which is a pretty good metric for looking at evolutionary change. So here, kind of a similar story. The values are actually inverted because we have to normalize by the number of generations. So if we look at the average number of generations between coalescence events, we see that it's a very, very low average number of generations in non-plastic populations. They are constantly having these selective sweeps compared to static and plastic populations that don't really differ from each other, but have many more generations between these sweeps compared to the non-plastic populations. So overall, all the different metrics, and I only mentioned a few of them here in this talk, we see that adaptive phenotypic plasticity slows evolutionary change in these fluctuating environments. So that brings us to the second question, which is, does adaptive phenotypic plasticity facilitate the evolution of novel tasks? So to start to get into this question, we looked at the same fluctuating environment that we used for the first part, but all we needed to do, we still have these environment A and environment B that are cycling back and forth, but we add 71 additional tasks, these novel tasks, on top of that. So these are always rewarded, but they have a much smaller reward value. So if you can perform one of the base tasks, that's better than these novel tasks. But if you can perform them, that's great. That's going to give you a little advantage over the other organisms. So we can look at the final or the most abundant genotype at the end of phase 2 for each replicate and just say how many of these novel traits are actually performed by that most abundant genotype. And when we look at these, we see that non-plastic treatment, the non-plastic populations perform very, very few. Most populations are performing 0 of these novel tasks. A few populations might perform 1 or 2, but very few compared to the static and plastic populations that are performing, at least a good handful, right? They're not performing all 71 by any means, but they have, a pretty decent chunk of the tasks that they're performing. So this raises some questions of like, is the static and plastic or are the static and plastic populations, better at exploring the fitness landscape and finding these novel tasks? Or, is there something else going on? So we dug into this by looking at how frequently those tasks are discovered along the different treatments. So when we look at these, we actually see that for the most part, these treatments do not differ in how often they discover these new tasks. The only significant difference, which is weakly significant, is between static and plastic, but non-plastic here is no different than plastic and static treatments in terms of how often it discovers these new tasks. So the missing piece of the puzzle then was looking at how often these novel traits are lost along the lineage. So when we look at this loss rate or loss frequency, we see that static and plastic populations very rarely lose the novel traits that they have evolved, which is very different than the non-plastic treatment that is almost constantly losing, it's evolving these different tasks, but then it's losing them. So that's why we, at least we expect, that's why we're seeing pretty much 0 novel traits at the final, in that final organism, final genotype. So for this section, the takeaway, I guess, is just that adaptive plasticity doesn't necessarily change the task discovery rate for novel tasks, but it does help you retain those novel tasks in this cyclic environment. And then last but not least, which is new because I didn't get to talk about this in the other talk, it was too short, is does adaptive phenotypic plasticity increase the accumulation of deleterious mutations? So we ask this question because in the theory, if you are a plastic organism and you're expressing different phenotypes, often that means that you're only expressing parts of your genome at a time. So in theory, if you're only expressing parts, the parts that aren't expressed can be getting hit by mutations, and that's not actually affecting your phenotype until the environment switches later on. So there's a potential that these deleterious mutations are accumulating. So we wanted to test this. And again, we went back to the same fluctuating environment, but this time we added this deleterious instruction. So this is explicitly deleterious. All it does is reduce your fitness, and it has no other purpose, at least by itself. So if you're performing this, it's going to hurt you. There's no way around it. So we looked at the percentage of replicates where this deleterious instruction was executed by that final most abundant genotype. And we see it executed in 14% of non-plastic lineages, or populations, I should say. But we actually see it in 0 of the plastic and static populations. which is interesting, right? So it looks like non-plastic populations are executing this, are accruing more deleterious mutations than the other treatments. And we decided to look at this, maybe it's like the last question where it's a factor of that very last generation. Maybe we should look at it over time. So if we look at how often this deleterious mutation is coming in across the lineage, we see very few of these mutations popping up in the static and plastic lineages, but we see, a considerable amount of them appearing along the non-plastic lineages. So overall, it seems like deleterious mutations are actually more common in non-plastic lineages in this cyclic environment, at least in our experimental setup. So to try to put a bow on top, compared to non-plastic lineages, adaptively plastic lineages, 1, experience slower evolutionary change, 2, better retain novel tasks, and 3, accumulate fewer deleterious mutations. And, oh, my slide changed, whoops. And one thing that I mentioned kind of briefly, but I didn't actually dive into, is most of the time on pretty much all of our metrics, the plastic lineages are appearing much more similar to the static lineages than to the non-plastic lineages in that same environment. So overall, and this is why the title slide had spoiler in front of the title, all we're seeing is that this plasticity, this adaptive phenotypic plasticity, is really just stabilizing evolution in this fluctuating environment. All right, and there's tons of future work we can do. As always, we use the cyclic environment, which was great and easy, but there are so many hypotheses that we wanted to test that you just can't, because they require things like novel environments to be encountered by plastic organisms. And we, went between A and B, A and B, A and B, so nothing was ever novel in terms of the environment. And of course, there are all kinds of different ways we can tweak the environment. One thing that I would love to do, or see someone else do, is to try this in something other than a beta, because the cryptic mutations, the mutations occurring in the unexpressed part of the genome in plastic organisms, is fairly unlikely just in the way that a beta organisms work and how they evolve. But maybe if you tried in a different system, you will see a lot of that cryptic variation, cryptic mutations, where deleterious mutations are accruing. So if somebody wants to do that, I would love your paper and cite it all the time. And then finally, of course, we can look at other forms of plasticity that aren't necessarily adaptive. But that's it for me. Thanks again for having me. I'm honored that you all like the paper. So I wish Alex could be here, but hopefully that was an okay talk.
Are there any questions?
Yes, hit me up with all the questions.
I've got one.
Yeah, absolutely.
First of all, great talk, very interesting, and actually it was helpful to hear it twice. So it's sort of a multi-part question, but it's all focused in one area. So in your phase 1 and phase 2, first of all, just to make sure I have this right, the cyclic environment was applied that whole time, phase 1 and phase 2.
Yeah, whatever treatment we're looking at, if it is one of the cyclic environments, it is, it's exactly the same. And same for static, we just carry it over.
And so you started off with organisms that are not plastic, but in phase 1, they evolve the plasticity. And then at the end of phase 1, you've got it, and then you test it.
Yes.
Okay, so what I don't think you said, what exactly are the phenotypes, and did you actually just observe, okay, here's how it behaves in environment A versus environment B? What is that?
Yeah, that's a great question. I didn't think about that, but I should have definitely touched on that. So our phenotypes here are just which of those tasks are performed in each environment. So for example, at the end of phase 1, we actually made sure that the organisms in the plastic treatments were perfectly plastic, which meant that in environment A, they performed those 3 tasks and not the other ones. And then environment B, they only performed the 3 that were rewarded there. So really, when you look at both environments, and look at which tasks are performed in each environment, that was our phenotype. I hope that makes sense.
Yes, and maybe I'll extend that question a little. So in the other 2 treatments, I think what the non-plastic treatment is really an organism that is exposed to a cyclic environment, but doesn't have the capability to adapt to it, really.
Absolutely.
And whereas what did you call it? The static environment, they evolve. It's interesting that the static and the and the what did you call it? The plastic are so
Yeah.
similar across all the measurements. They're pretty much the same. So
Yeah.
basically, because they're both well adapted, I guess.
Yeah, absolutely. Absolutely. They don't have to change anything to keep up with the changing environment, right? They're good, more or less. And then we can throw additional tasks, and stuff like that in on top. Yeah.
But if you put, I mean, this is a possible additional experiment, but I think we can probably all predict what would happen. If you put the static individuals then into a fluctuating environment, they would probably be at quite a disadvantage.
Yeah.
You put the plastic individuals into the static environment, they would probably gradually lose their plasticity over time.
Absolutely. What is it? Genetic assimilation? Is that the term? I always forget the term, but I don't know if it's no, it's not assimilation. I always forget the term. But where it's, they do, you're totally right. Canalization, I think is the word, right? Where they do lose their plasticity. But yeah, I think your predictions there are spot on. And specifically, if we put the static populations in the fluctuating environment, I think you're totally right, where they would struggle. Other than where they are performing all the tasks, if they get lucky and mutations turn off the right tasks, they will adapt probably considerably quicker than like the ancestral organisms did. But yeah, no, I totally agree with all of your predictions.
Okay. Thank you. That's great.
Yeah, thank you.
Are there any other questions? So,
all right. Well, thanks all for having me. Really appreciate it. And again, honored that you liked our paper. So now I'm excited. I get to kick back and watch the other talks. So, all right, I'm going to mute and turn my video off and let you all continue.
okay, thank you for your time, Austin. So I think, so it's 8:30, I think we can move on to the lightning talks. And the first lightning talk should be given by Atusa Parsa, who is a PhD student at the University of Vermont on the evolution of computational granular metamaterials. So Atusa, if you want, you can just screen share and start your lightning talk.
Yes. Hi, everyone. Let me just share my screen. Okay, can you see my slides okay?
Yeah.
Okay. So thank you so much for the introduction, Federico. As Federico said, I'm a PhD student at the University of Vermont. And today I will be talking about my work on evolving computational granular metamaterials. I'm going to start with an overview of my work. In this work, we present granular metamaterials as a promising choice for the realization of mechanical computing devices. And we demonstrate the design of basic logic gates embedded in granular metamaterials. And we also show that the design material can be reprogrammed by frequency modulation. So here in panel A, you can see an overview of the multi-objective optimization process. And in this framework, each individual in the population is a granular metamaterial that's composed of two types of particles that are shown in different shades of gray in this figure here. And so different configurations of these particles confer different material behaviors. And the objective here is to design materials that can act as logic gates. So in order to do that, we need to measure the computational functionality of a material. And so what we do is we apply vibrations with 0 or non-zero amplitudes to the input ports and measure the amount of vibration at the output ports. And our results confirm that we can design materials that can act as an AND gate, as you can see in panel B, or as an XOR gate in panel C. And we also show that multi-objective optimization can discover a single material that can act as an AND gate or an XOR gate, depending on the frequency of the input. And then we showed how the superposition of the input waves that are supplied to the system will result in emergent behaviors other than what the material was originally designed to do. So for a single material, we have two binary inputs. And so we have 4 different cases that are shown in 4 different colors in these figures. And I'm going to go through one simple example just to show you how the material behaves in each of these 4 cases. So the first case is the 0, 0 case. If you follow the orange outline in this figure, so this is the case where no vibrations are applied to the input ports. So the particles with green and blue outlines are standing still. And as you expect, because no vibrations are applied to the material, we see no vibration at the output. And so the output is also 0, which is consistent with the functionality of an AND gate. And next is the 0, 1 case. Here, I hope that you can see the movie here, that the blue particle is oscillating. So we apply a sinusoidal wave to the blue particle, and the green particle is standing still. And as we expect, the material that's evolved, it's blocking the propagation of this acoustic wave through the material and to the other side. And so we have a 0 again at the output, which is again consistent with the functionality of an AND gate. And next one, it's 1, 0 case. This time the green particle is moving, the blue particle is still. And again, we have a 0 at the output. The last case, the 1, 1 case, if you follow the yellow outline, you can see that both of the input ports are oscillating, and we are applying sinusoidal waves to both of the input ports. And this time we can see the green, the red particle starts to oscillate, which indicates a 1 at the output. So this was just a simple example to show you how we expect the evolved material to behave and what we expect from the material. Let's take a step back and talk about why this research is important, why we want to do this. Here in this figure, you can see the hierarchy of a computing device. There are 3 different layers in this figure. The top layer is the computing model, which is just a mathematical model of how the computational process unfolds. Then we have the engineered architecture here, which is the abstract platform to implement the computing model. And then the last layer, which is what we are focusing on here in this research, is the physical substrate. And so traditionally, an engineered architecture is realized in digital electronics substrate. But this hasn't always been the case. The idea of mechanical computing devices has actually been around for a long time. And throughout the history, there has been a number of mechanical devices that has been used to perform some sort of computation, as you can see a number of them here. And at some point, electronic forms of computation overtook the mechanical forms because they had better potential for miniaturization and integration. But recently, with the development of new technologies and new materials, it has opened up the door to new potential physical substrates, and metamaterials are one of the promising ones. So in this work, we proposed the usage of granular metamaterials as a physical substrate to do computation. So what are metamaterials? Metamaterials are engineered composite materials, and specifically granular metamaterials are materials that are composed of these discrete particles. And so there are a couple of advantages for these materials because they are made of these discrete particles, and the particles can be reactive to different environmental conditions, for example, based on the temperature of the environment, or for example, the magnetic field in the environment, they can change their material properties. So we say that they can be dynamically programmed. And there are many parameters in the system that can change the response of the system. But you can imagine that with so many parameters to choose from, and because of the complex relation between the structure of the material and the behavior, they're really hard to design. And usually traditional approaches, which are based on trial and error, they fail in this high dimensional space. And so in this work, we use evolution algorithms to automate the design process. So here is an overview of our system, and it's a real simple system. It's a 2-dimensional assembly of 2 types of particles. Some of them are hard particles, some of them are soft particles. and they are placed on a hexagonal lattice. We choose two particles on one side as the input ports, and we choose one particle on the other side as the output port. And so we want to make this material act as a logic gate, where the inputs and the outputs are acoustic waves. So we apply sinusoidal waves to the input ports and measure the amount of vibration at the output. So when a vibration is applied to a particle, it causes displacement from its initial position, as you expect. And we need to come up with a bit representation for this material because we want this material to act as a logic gate. So one possible representation is to use the amplitude of the displacement signal as a bit abstraction, where you can see the truth table here, meaning that if there is a vibration at the output, it indicates a 1 at the output, and if there is no significant vibration, it indicates a 0. So there are 4 different cases, and for each of these 4 cases that are shown in this table, we can calculate a gain, which is defined here, and it's simply just the fast Fourier transform at the driving frequency at the output port divided by the amplitude of the fast Fourier transform at the input ports at the driving frequency. And so based on this, because the goal is to design an AND gate and also an XOR gate, we need to design 2 objectives. These are the 2 objectives that we defined. For each of the cases, for a logic gate, we want the relative magnitude of the gain to be consistent with the desired functionality of the gate. So for example, for an AND gate, we want to see a higher amplitude of oscillation at the output when both of the input ports are excited. So we want to see a higher gain, a higher G11, and we want to see a low amplitude when only one of the ports is driven by a sinusoidal wave. So that's why we chose these 2 different objectives. And then just to recap.
It's a great talk, but you're running over time, so please keep it short.
Oh, sorry. Yeah, sure. Sorry. So let's just move on to some results. So here in panel B, you can see the progress of multi-objective optimization, and we chose one of the individuals from the last generation to examine its functionality. And here you can see the functionality of that candidate solution. You can see that we can program the material by modulating the frequency of the input signal. For example, in panel A, you can see that the material is acting as an AND gate. In panel B, it's acting as an XOR gate at another frequency. And we also show that we can program the material to act as a half adder. So the material wasn't initially designed to act as a half adder, but the point in this research is that we can take this a little bit further and program the material by regulating the input frequency so that the material can behave differently. To conclude, in this work, we showed the potential of the granular metamaterial to act as physical substrates to embed some sort of computation. We showed the multi-objective optimization process, and we showed that we can find some materials that can act as an AND gate or an XOR gate based on the frequency of the input. And we showed how we can reprogram the material using frequency modulation. So in the end, I just wanted to introduce my collaborators, and thank you so much for this opportunity. I'll be happy to answer any questions. Thanks.
Great. Are there any questions? We have time for a couple of questions.
Yeah, I had a question. Sorry. I wondered, so you see that you have 2 positive inputs, but I was wondering if you could do things like a NOT gate, where obviously it seems like you can't create something from nothing. So then would you have to have a motor or something like that? Have you done any thought about that?
Yeah, that's a great question. And actually we are working on that. So in order to do something like a NOT gate or even an AND gate, you need to have some sort of a power source in the system, right? And we are working on it right now. We have some positive results. What we do is we just have one particle that's constantly vibrating. So the material will be like blocking the propagation of that vibration from the source to the output based on the material, the position of the different particles. yeah, that was great. Thanks.
Okay, great. So thank you again for your presentation. I think we can move on to the next speaker. So the next speaker is Georgina Montserrat Resendiz Benumea from University of New Mexico, and she will talk about initial steps toward development of minimal affectivity in collectivity models under a 4E cognition perspective. So the floor is yours.
Thank you. So I'm going to share my screen. Is it fine?
Yeah.
Thank you. Okay. So, okay. So thank you for this kind of presentation, Federico. And well, welcome to my presentation. My name is Georgina Montserrat Resendiz Benumea, and I am currently collaborating with Dr. Jesus Mario Siqueiros, Dr. Carlos Gershenson, Dr. Gabriel Ramos Fernandez, and Dr. Katia Rodriguez Vasquez. I am a PhD student at IMAS UNAM from the National Autonomous University of Mexico. And well, our presentation for the ALife 22 era workshop is the following: initial steps toward the development of minimal affectivity in collectivity AIC models under a 4E cognition perspective. So the contents of this presentation are the following: introduction and background, then our proposal, and finally some of the references. So we're going to start with the introduction and background. Well, first of all, this work and this proposal was submitted to the 4th International Workshop on Agent-Based Modeling of Human Behavior, ABM-HUB 22, as an extended abstract with title, Paving the Way Toward Minimal Affectivity in Collectivity, AIC Models, a 4E Cognition Proposal. And it is going to be presented on Thursday, July 21st, at 14:10 to 14:20 UTC, and we will be very happy if you would like to join us to this presentation as well. Well, now the background of our research. Well, traditional cognitive science has been mostly dominated by a computational paradigm in which it is assumed that brain is a computer, cognition is computation, and behavior is a mere product of neural activity. In stark opposition to this view, we have the 4E cognition approach, which comes from embodied, embedded, enactive, and extended, which in conjunction argues that the basis of cognition can span over brain, body, and environment. In this sense, social interaction can play a role as an enabler of cognition, thus enhancing our individual capacities. In order to demonstrate these theoretical groundings, some simulation models have been developed as computational proofs of concept. As an example of these models, which are also known as embodied dyadic interaction models, we have the Kandari et al. 2019 model, which is a minimal model of cognition based on evolutionary robotics. And with this model, they demonstrated that social interaction increases the complexity of neural and behavioral dynamics of embodied agents and revealed that this cannot be achieved in isolation. To this model, they used pairs of acoustically coupled agents in which each of them has one acoustic emitter, 2 acoustic sensors, and 2 motors to move in a empty bidimensional environment. Now we have here the experiments of the Kandari et al. 2019 model. They consisted of evolving agents in 2 main scenarios. The first one, the interactive scenarios, in which each of the agents is responsive to the other, so we can have a bidirectional interaction. And the second one, that is the isolated scenario, in which we evolve agents without the presence of a partner. So the goal of these experiments was to maximize the neural entropy of the active agents. And in the first scenario, what they found that the strategy that the agents followed was by mutually interacting. By doing so, they increased both neural and behavioral complexity. While on the other case, when they had the isolated scenario, by maximizing the neural entropy, they found a loss in both neural and behavioral complexity in comparison to the case of the interactive scenario. Now, they tested the best pairs in the interactive scenario under what is called the ghost condition. So one of the agents in this case, we can see the light blue agent is the ghost partner. So it is only repeating behavior from previous trials. So it is not responsive to the active agent, that in this case is the red one. So in this case, we have only unidirectional interaction. And by measuring the neural entropy of the active agent, that is the red one, they found that there is a loss as well in both neural and behavioral complexity in comparison with the interactive scenario that was the original one. On the basis of these studies, we developed a new one with title Shunken Social Brains, a minimal model of the role of social interaction in neural complexity. And in this model, we demonstrated that smaller brain agents, that in this case is a 2-neuron model, evolve in social conditions, can achieve comparable levels of neural complexity as bigger brain agents, that in this case is a 3-neuron model, evolve in solitary conditions. As an example of the results, we have here the agent behavior, in which we can see that the 3-neuron model in individual evolution have a simpler behavior, only simple circles. And in the other case, we can have the smaller brain agents, that is the 2-neuron model. In social evolution, we can see that both agents are creating a more complex behavior in this kind of coordinated loops, as we can see in the second image. Right. So what is our proposal then? Well, on the basis of this work, we propose to integrate the phenomenon of affectivity in its minimal way. So we have that from a 4D cognition perspective, we have that agent environment interaction and affectivity are constitutive to cognition. Now, in the field of social cognition under a 4D cognition perspective, we have a social interaction. In the case that we have been working on these embodied dyadic interaction models, and we're planning to integrate this phenomenon of affectivity and integrate both of them in our models that we call them the minimal affectivity in collectivity, AIC models, in order to study the role of affectivity in social phenomena. Right, so how are we understanding affectivity? Well, affectivity, we understand it as agents being sensitive to the precariousness of their dimensions of embodiment. And by dimensions of embodiment, we refer to the organic, the sensorimotor, and the intersubjective. In our previous studies, we have been working mainly on the intersubjective and sensorimotor dimensions, but we haven't worked on the organic one. So that is why we are planning to integrate it by doing a transition between acoustically coupled agents to light-powered agents with an internal battery, to light sensors, to motors, and a light emitter. This is to simulate a minimal form of metabolism. And also by doing some evolutionary experiments as the ones that we did with our other models, and also to test them under a social evolution condition and an individual evolution, and then compare in terms of neural complexity with the experiments that we did with acoustically coupled agents. And well, more details are going to be given in the ABM workshop. And well, some of the references here. We have here some of the work that we have done in embodied dyadic interaction models. And this is my email in case you would like to contact us. And well, that's all. Thank you very much.
Thank you. So are there any questions from the audience?
I have a question. So forgive me if I overlook this, but you mentioned that you were measuring behavioral complexity in your experiments. Is that correct?
Well, for the moment, we're only just as a visual kind of behavioral complexity, and we're trying to find some of the some kind of measures for example, for this picture. we're trying to find which could be a good measure for this kind of behavioral complexity. But right now, the measure that we're mostly working on is on neural complexity by using neural entropy and with Shannon entropy.
Sure. Okay. And then I guess maybe not, because you mentioned that it was a minimal model, but I wonder if there was any environmental complexity. So was there a physical environment that these agents were interacting in, and what was the complexities associated with that environment, if there were any, or was it just a blank space where two agents were interacting with each other?
Yes, well, in our last study, it was just an empty environment, but previously we created another environment in which we evolved these agents with ghost agents, just to have some kind of non-interactive stimulation. But in our last study, it was just an empty environment, so it hasn't any kind of environmental complexity. Yes.
Okay, great. Now I'm just curious, and what is sort of your ideas for the future direction of this? Do you have any immediate next steps that you're looking to pursue?
Oh yes, well, the main thing of these studies is to mainly work on the role of affectivity in social phenomena. So we're pretty interested in understanding this kind of behavior, like the ones that is shown in, for example, swarms. So we want to understand how affectivity in this minimal sense from a foreign cognition perspective, and mostly in the sense of precariousness, how a precariousness is affecting this kind of social behavior that is presented, for example, in ant colonies or in humans, and for example, groups of primates and all of that. Yes.
Cool. Oh, thank you.
Thank you.
Thank you for your talk. So I think we can move on to the next speaker. So the next speaker is Fernando Rodriguez, and he will talk about his phenomenological experience, a cognitive phenomenon. I hope Fernando can screen share.
Hi.
Hello.
Hi. No, it's not the
All right, so the random sample kind of
I don't know why. Okay. Well, hello everyone. Mine is really very simple. Wait, I can't really see here. Okay. So my question basically is there some kind of relation between phenomenology and some kind of cognitive property? We normally have assumed since Varela, I mean, basically since the embodied mind, this, right? That starting with life or the autonomy of life, we have some kind of meaning, in a minimal sense at least. And the more traditional approaches in cognitive science, normally related to agency, they have kind of worked based upon these ideas, right? And trying to fill the void from the original presentation of what autonomy was, with dynamical systems and more complex concepts like normativity, adaptivity, and others, right? The problem is it doesn't seem to work as we were expecting. We have, I'm just citing these three here, but if you want to see it like this, is evidence from different points of view, trying to say the same, that maybe it is not true, or there's not really a kind of proper justification to say that autonomy of the living or autopoiesis is enough to justify in cognitive terms the emergence of what we call phenomenology. So some alternatives that have been proposed are this one. Maybe I should have also considered here radical inactivism. They say that is culture. Here they say that is language. Maturana has always, or was always, very kind of emphatic about the idea that an observer can only begin with language. There's no kind of previous kind of self-reflective. I think that's very discussable, but I'm not really going to talk much about that. Then we have this proposal, which I find very interesting, but is very far-fetched from a simple kind of simulation. I mean, that's the thing that I work with. So for minimal systems, I don't really see how I could do it, at least not for now. And this one, which is integrated information theory. And this is the one that I'm working with. Basically, very roughly, what they propose is somehow similar to, they have some kind of, it's not really, you cannot kind of create a map of ideas, but you can find some interesting kind of links between what we understand of autonomy, like the intrinsic existence, a causal structure of mechanisms as an emergent system. And what the IIT proposes is that if you fulfill the properties that they give as necessary for having consciousness or phenomenological experience, you should be able to characterize it arising, right? So what I'm doing is I only have preliminary results because the computational processes are super expensive. But basically what I'm doing, I have taken the glider idea of Randall Beer, and assuming that is a simulation of an autopoietic agent, or at least an autopoietic system. And because in Randall Beer's analysis, you can see a lot of cognitive properties, which emerge from the very fact that there's a kind of an autonomous organization behind it. And you can see a lot of the cognitive properties, but we're not really able to see any kind of thing relating to experience, right? So what I'm doing is trying to see if you can measure on the same glider intrinsic information, which loosely can account for the existence of a phenomenological domain, and integration, which not really loosely, I think it's going to be really useful. It can account for the idea of emergence. The so frequently used idea that an autonomous system is emergent unity, right? So the results that I have until now, they show that when the glider moves, it's not really a thing that is moving, right? It's just a recursive network of processes creating and destroying and maintaining some cells, right? And the interesting thing that I found is that when the glider creates new parts of the glider by taking cells that weren't part of the glider before, it kind of, you can obtain high values of intrinsic information. Intrinsic information understood in this sense as kind of what the agent sees from its point of view. That's basically the idea. I still have to measure the integration, but that is super, super expensive because you need to do all the computations for super heavy. For those of you that know about the integrated information theory, probably are going to understand. And that's all really for now.
Okay, thank you for your talk.
Thank you.
Are there any questions from the audience?
I maybe have a small question. We've been recently talking about similar things like this, where we try to make simple physical models, like a real model systems with droplets. That do similar stuff as gliders even. And we kind of want to use that to show that maybe minimal chemical physical systems can have some sort of cognition or something like that. And we were kind of struggling with the thought of like, how do we differentiate? And maybe this can help between, let's say, a ball falling down, a bowling ball falling down to earth, and a droplet that moves on a water surface, but just because it moves, like it dissipates energy and it has like a behavior because of that, and that behavior increases its survivability or something like that, or it's like the glider can find new materials to make new gliders.
Yeah, I think that's a very, very interesting thing, you know. So for example, if we believe in the idea of autonomy, basically what we're accepting is that dynamical transitions of the system kind of have some kind of recursive nature, right? But we don't know actually what are all the states of the, like, belonging to the organization, right? And the thing is, when you see the glider, the distinctions that Beer makes about what is a glider and what is not a glider are heavily kind of perceptually guided, you know? And I think the interesting thing here of introducing this idea of intrinsic information is that it gives you the possibility to know if some configuration of the glider in this case, or maybe in another case, a different thing, can be counted as a part of the organization, you know? Maybe even if it doesn't really look as a glider, you know? I think that is, I think that's a kind of, because on the one hand, you have the idea of transitions between states, and the IIT kind of introduces the idea of telling you if this state is actually, I don't really believe, this is my personal opinion, that the IIT is really describing consciousness, but it's actually describing like an emergent system, you know?
Okay, yeah, interesting thoughts. Yeah, I would, yeah, as Austin also says, it will be interesting to see the full work. But as you said, it may take some time since it's expensive.
Yeah, I mean, I first kind of replicated the system that they present in the IIT paper, and it took me a while. One, because not all the math is really explained, and also because it's kind of heavy.
All right, thank you.
Yeah, thank you.
Oh, great, thank you for your talk. I think you can move on to the fourth and last talk for today, which will be given by Stavros Anagnou. Hope the pronunciation is correct, from University of Hertfordshire, on towards an emotional model of social norms. So Stavros, the floor is yours.
Hi, thank you very much, Federico. The pronunciation was perfect, actually. And yeah, thanks for organizing this workshop, and to the organizers of ERA. I really enjoyed all the talks so far. Let me just share my screen. If there's any barking at any point. There's a loud dog, so I'll try to manage with that as best I can, but I think it's okay for now.
I think you have to post a picture on Pet Parade on Slack.
All right. Okay, I will. All right. Can you all see and hear me well? Okay, let me just start my timer. Right. So this is sort of a very broad overview of sort of what is my PhD. I've done some work already, which is more specific and put out there, but I think I kind of want to give you this kind of general flavor and then start conversations from there, particularly relating to conceptions of affect. If you have any takes on that, I'd be very interested to hear them. The more viewpoints, the better. So yeah. And for context, I'm a second-year PhD student at the University of Hertfordshire. So, social norms. What are they? So I take the minimal definition that they're group-level behavioral regularities. They're useful in all sorts of ways for collectives because they allow for cooperation. So say a norm around not stealing, being more altruistic with resources. They're also useful for coordination and communication. An example being on which side of the road to drive on, which is also important for large-scale collectives. They're also especially important in sort of the grand evolutionary scheme of things because they extend cooperation, or they're one of the mechanisms that extend cooperation beyond just being altruistic with your genetic relatives, which means that, you know, social norms and how they're enforced might be one of the sort of building blocks of large-scale society. So yeah, very important. And they're maintained through strategies like sanctions, which sanctions, although recent events have made us think of sanctions in a very specific way, sanctions in sort of the norm literature are a general catch-all term for punishments, maybe physical or non-physical, and things like shunning and ostracization and stuff like that. So, although there's been loads of work, especially in the evolutionary game theory community, on how social norms are implemented, what kind of strategies ensure they emerge. You know, one strategy that ensures the emergence of a cooperative norm in evolutionary game theory is tit for tat. You know, the golden rule: if someone's nice to you, be nice back to them. If not, the other way around, defect against them. And the aforementioned sanctions, the punishments when someone violates the norm, will ensure the emergence of the norm, whatever it is. So although there's been lots of attention given to that, and I won't refer to it for the sake of time, less attention has been given to how these strategies like tit for tat or sanctions are implemented in biological systems. And generally, we sort of feel our way through the social world. We punish someone because we are angry, and that motivates us to punish them. In a situation where we are uncertain of what the social norm is, like the picture down here with the very worried looking man. If anyone recognizes the experiment, extra points. We start becoming if we don't know what the social norm is, we get very anxious and start being very vigilant and looking around us at what others are doing. So I think if you want to have a full model of social norms and how they emerge, we should also incorporate affect into multi-agent simulations, which is the general thrust of my PhD. So to do this, if we want to incorporate a model of affect into the agent architecture, there's many of them that exist. I'm going to take one for now and stick with it, but I'm open to changing in future, of course. Since this is I'm preaching to the crowd here, so using an ALife sort of embodied conception of emotion. So I don't model it explicitly. It's not a box that says angry with arrows going into it and then arrows coming out. I see it as a sort of emergent bottom-up phenomena, dynamic patterns of hormonal modulation. So basically, I look at emotions as these dynamic processes that coordinate things at multiple levels of organization. So that's physiological, motivational, perceptual, etc. Processes to maintain homeostasis and promote survival. That's the key aspect of emotions here. So just a quick example in the context of social norms to give a flavor. Scarce resources, sort of environmental influence, might lead to the release of a stress hormone. which this I want to emphasize the stress hormone. I'm not saying that's emotion. I think it's just part of the sort of the causal chain. So a stress hormone is released. This, one of its functions among many, might be to increase vigilance to norm violation, for instance, stealing, because this is especially important when resources are scarce. And when resources are scarce and people are stressed, you might notice that they get more irritable. And this may increase the action tendency for them to carry out an aggressive punishment, which would enforce the norm. So yeah, that's just a very general idea. I'm happy to take questions on that at the end. I realize this is a lot in one slide. But yes, I kind of want to incorporate this into a model of social norms to see sort of the biological sort of underpinnings of how social norms emerge, what mechanisms underpin the emergence of norms in biological systems. So what do I think the benefits of this approach are? So as generally we see with formal modeling, it allows for what Di Paolo, who was mentioned earlier, called, along with colleagues, opaque thought experiments of various hypotheses about biological systems, and you're forced to make all your assumptions explicit and make your argument in very explicit ways, so it can be compared to other arguments and so on. This allows a systematic exploration of how emotions play a role in social norms. So that's why I think they should be formally modeled first, as well as empirically investigated. Also, the multi-agent models we see in ALife and other disciplines are really good for looking at the sort of group level regularities that emerge from the individual level behaviors, which in this case will be motivated by social norms. Certain other aspects of social norms, or rather of affect, which are actually interesting to, and emotion, that are good to explore with this kind of simulation. Affect, in this case, I have an example here, the oxytocin hormone, can sometimes result in positive feedback loops. So you have promotion of social interaction through this hormone oxytocin, which leads to more release of oxytocin, which leads to more social interaction. And those of us who knows a bit about complex systems, this can result in quite complex emergent phenomena, which are best explored through agent-based modeling, among other things. So I'm tight on time. I'll just end on a final point. Since I'm looking at taking a bottom-up perspective, I also think this is a way to naturalize the study of social norms and affect, because we're going to be looking at mechanisms that could be present in animals. And we could also see how social norms emerge in animals and get away from the sort of very cognitive, rational view of social norms. So yes, that's my talk. Thank you very much. Any questions?
Thank you. So are there any questions from the audience?
Yeah, I have a question. I think the topic is super interesting, but do you think there's which do you think is kind of the minimal level in which you can kind of study this emergence of social behavior, you know, in terms of in terms of cognitive complexity?
So I think in terms of the most minimal model you can get, you don't have to necessarily invoke affect, like I am here to do that.
Mm-hmm.
The project I'm doing isn't so much an origins of social norms in general, which I guess would be the more ALife type thing
Mm-hmm.
to do. I'm look specifically the explanatory targeted social norms from the from like how do you get from affect to social norms, and then reason how maybe this occurs in use the models to guide explanation of how this occurs in biological systems. I think it can be done in a more minimal way, for like, you know, using Bayesian agents or some of the approaches that I mentioned earlier as well. But yeah,
it's just because I saw the citation from Lola Cañamero.
Yeah.
No, yeah, okay. Thank you.
Is there anything specific you want to say about?
No, because
okay.
she normally works kind of a very specific kind of how would you say, a cognitive level kind of middle, in the middle. So it's not very,
Well,
it's not a minimal level, but it's not really also a higher like human level necessarily.
Oh, I see. Yeah, I guess technically it's bottom up compared to the other approaches I've seen. But I guess it technically, with regards to really minimal approaches, it's middle out, I think is the term.
Okay.
Yeah. So yeah, thanks for getting me to clarify that.
Thank you.
I appreciate it.
So thank you, Stavros, once again. I think time is over, and I would like to thank all of the presenters today, the invited speaker, as well as my fellow ERA board members for organizing this workshop. If you're interested, there is going to be an extracurricular ERA social event on Thursday at 6 p.m. CT, which is more a more laid back event with the academic karaoke and social stuff going on and so on. Thank you so much for participating.
Thank you.
Thank you very much. Everything was really well done. Cheers.